Chapter 12, Section 4, Gene Regulation and Mutation. The main idea for this section is gene expression is regulated by the cell and mutations can affect that expression. Some keywords to take note of is gene regulation, operon, mutation, and mutagen. Prokaryote gene regulation is the ability of an organism to control which genes are transcribed in response to the environment. An operon is a section of DNA that contains the genes for the proteins needed for a specific metabolic pathway. So a few things an operon contains: an on and off switch, a promoter, which is where RNA polymerase binds, a regulatory gene, and genes for coding proteins. So in this module, you see the Trp Operon. Tryptophan synthesis occurs in 5 steps controlled by the Trp. The Trp Operon is a repressible operon because it is usually repressed or turned off. The LAC operon. When lactose is present, E. coli can synthesize an enzyme to use it as an energy source. And the LAC operon is an inducible operon because an inducer binds to the repressor and inactivates it, allowing transcription to be turned on.
Gene regulation is the ability of a cell to control which genes are expressed in response to its environment. Let's look at how this works in prokaryotes. In prokaryotes, gene regulation is often controlled by an operon. An operon is a section of DNA which contains the genes needed for a specific metabolic pathway. The operon is made up of the promoter. This is the segment of DNA that is in front of the genes, coding for proteins needed in the metabolic pathway. The operator. This is a part of the promoter where RNA polymerase binds and starts transcription. The regulatory gene. This is usually located before the promoter, and it's a gene that codes for a regulatory protein that keeps the operon on or off. The gene or genes coding for our proteins. An operon can function in one of 2 ways. A repressible operon represses or decreases transcription. An inducible operon can induce or turn on transcription. An example of a repressible gene is the Trp Operon. That's the one shown here. The Trp Operon is a cluster of DNA that regulates 5 enzymes necessary for tryptophan synthesis. The transcription of the Trp Operon is usually turned off. After all, if you already have tryptophan, you don't need to make more. In the off position, the repressor protein is bound with tryptophan. This changes the shape of the repressor protein, so it can bind with the promoter and block RNA polymerase. When tryptophan levels are low, you need more. The repressor protein has a different shape when it's not bound to tryptophan, so it can't bind to the promoter. The RNA polymerase is able to bind to the operator, turning the process of transcription on. This starts the process of transcription, and the synthesis of the 5 enzymes needed for tryptophan synthesis can begin. Now, let's look at an example of an inducible operon, the LAC operon. Like the Trp Operon, the LAC operon also contains a promoter, operator, regulatory gene, and some protein genes. The regulatory gene makes the regulatory protein that binds to the operator when lactose is not present. This prevents the transcription of the gene's coding for the enzymes needed to digest lactose. When allolactose, an inducer found in foods that contain lactose, is present in the cell, it binds to the repressor protein. This causes it to change shape. The repressor protein is no longer able to bind to the promoter on the DNA strand, so RNA polymerase is able to bind to the promoter and begin transcription. This results in synthesis of the 3 enzymes necessary for the digestion of lactose. Everything the cell needs to regulate those genes is right there. Pretty convenient, huh?
Read the following about controlling transcription. A permanent change that occurs in a cell's DNA is called a mutation. Point mutation involve chemical change to just one base pair. Missense substitutions, DNA codes for the wrong amino acid. Nonsense mutation is where codon for amino acid becomes a stop codon. Insertion, deletion mutations, we have additions or losses of a nucleotide to the DNA sequence, and these cause frame shifts.
I'll be teaching about mutations. which are just changes in DNA. Different things can cause mutations, like chemicals. Sometimes they happen naturally. But to understand mutations in DNA, we're going to use a table to simplify things and some examples, but there's some background information you really got to know. First of all, when we really read DNA, we're reading different letters, like A's or adenines, U's or uracils, G's or guanine. Another one of these would be cytosine. Every 3 letters on a molecule called RNA has a complementary set of letters that will bind with it, like A would be U, U with A, G with C, and these letters, specifically every 3 letters, bring a very specific amino acid. And there's 20 different amino acids that are put in different orders to build all the proteins on Earth. So here again, these letters and the order of these letters are incredibly important. So mutation is a change in the letters. But to help us understand this, we're going to use letters that you're familiar with, the alphabet. Because you can think of the genetic code being a sentence that's read to make something. So if you read the sentence, the big fat cat ate the wet rat, that means something to you. So different types of mutations, for example, a missense, we simply substitute one letter for another letter. This happens in achondroplasia. Now the biz fat cat ate the wet rat. Well, that doesn't mean a whole lot. And it would also make the protein wrong or build something wrong because the sentence is wrong. We can also have a nonsense substitution where we cut out a big piece of the information. Now it's the big rat, which doesn't mean anything. So we're going to have a whole piece of the protein missing. We can also have a deletion, which we just delete one letter that causes a frame shift. But the frame shift, it changes all 3 letters all the way across. Now, back to our example, we're going to put different amino acids and create a completely new protein that probably won't function, just like this sentence doesn't make any sense. We can also just add a letter and cause a frame shift. Crohn's disease occurs in this way. We can duplicate things. We can write something twice that messes things up. And in Huntington's disease, a neurological disorder, every generation this even compounds. So understand the relationship between the letters and making a protein, and you should understand mutations.
Most people understand that a mutation is some kind of change. It is a change of genetic material, and more specifically, a nucleic acid. RNA and DNA, they're both types of nucleic acids. So how does a change happen? Remember that in DNA, the base adenine, A, goes with the base thymine, T, and the base cytosine, C, goes with the base guanine, G. And that's all good, but what happens if the wrong base matches up? Many things can cause an error like that. There can be external factors, like chemicals and radiation. Internal things too, like something goes wrong during DNA replication, during interphase. Interphase is a stage that prepares cells to divide during mitosis. And now while these things can increase the chance that a mutation is going to happen, it's important to understand that mutations, they're random. If a mutation is going to be a helpful thing for an organism, which is rare, very few mutations are actually helpful, it can't just will itself to get that mutation. An organism can't just will itself to get this.
So one way is protein folding and stability. Even small changes in the DNA code can cause genetic disorders. The change in one amino acid can change the sequence of the protein enough to affect both the folding and the stability of the protein. It can occur spontaneously. DNA polymerase can attach the wrong nucleotide. It happens, but this is rare and it's usually corrected. Certain chemicals and radiation called mutagens can damage DNA. For example, chemicals can cause mispairing of base pairs or themselves substitute for base pairs. High energy radiation can eject electrons from atoms within the DNA molecule, leaving behind unstable free radicals.
Not like X-Men either. And more about that when we get to natural selection. Many mutations are actually neutral in their effect, meaning they neither help nor harm an organism. And some mutations are harmful.
So we're going to talk about the different kinds of mutations. First, we're going to talk about gene mutations. So DNA makes up genes, and genes code for proteins that influence or make up different traits. So when DNA has changes, otherwise known as a mutation, then different proteins can be produced, and this can affect an organism's traits. So let's look at the gene mutations. So first, substitution. That means you have the wrong base matched. So instead of A with T, you could put A with G. Scandalous. You can have insertion. That means an extra base is added in. You can have deletion. That means a base is removed. Insertions and deletions have the potential to be especially dangerous because remember, in protein synthesis, we talked about how bases are read in threes. Well, if you add a base or remove a base, suddenly the number of bases total has changed, right? So if you read in threes, depending on where it happened, everything that's read afterwards has the potential to get really messed up. We call this a frame shift. Now, these were all types of gene mutations, but we also have something called chromosome mutations. Remember that chromosomes are made up of DNA and protein. They're highly organized, and they have a lot of genes on them. So all of the body cells in your body have 46 chromosomes. Human sperm and egg cells have 23 chromosomes. Well, changes can occur at that large chromosome scale too. So let's talk about these chromosome mutations. Just like insertion in a gene mutation, where bases got added, you can also have something called duplication in chromosomes. These are mutations where extra copies of genes are generated, so extra copies of that chromosome are generated.
And lastly, body cell versus sex cell mutations. Somatic cell mutations are not passed on to the next generation. Mutations that occur in sex cells, X and Y cells on the 23rd chromosome, are passed on to the organism's offspring and will be present in every cell of the offspring after.